Hello, good morning. Welcome to everyone. Okay, I hope everyone is fine. Okay, myself Sarita Ramana, as you all know that. Okay, now let's begin with the today's topic. Okay, today's topic is about the validation. What is validation? Why validation is needed, and when should we do the validation process? Okay, and about the types of the validation, we will discuss everything in a very detailed way. Okay, I will try my best to provide the maximum detailed information about the validation process to you all people. Okay, let's begin with the definition. Okay, what is validation? Okay, validation is defined as the document, okay, authorizing documentary evidence that proves that the following activities, process, method, okay, that will produce the product consistently, which will lead to the expected result. Predetermined requirements, they will meet the predetermined requirements. Okay, this is the definition of the validation. Now let's say why should we do the validation? What is the need of the validation? Okay, there is a very important role of the validation. Firstly, it is the requirement of the FDA. US FDA means Food and Drug Administration. Okay, under the current good manufacturing practice 21 CFR 211 and under the GMP regulation guideline 21 CFR 820. Okay, there is the requirement of the process validation. We will go under the types of the validation, but this is the requirement. Okay, so we need to carry out this validation process. And other importances are like as this will help us to reduce the cost. Okay, this will prevent us in failing in from the GMP. Okay, this will help to save the cost. Okay, and we can also say that it is a documented evidences that the process we are using in manufacturing the drug by the pharmaceutical company, that our products, our drug are qualitative and efficacious. Okay, so when should we carry out this validation process? Okay, validation is carried out as per the written protocol. Okay, as per the written guideline, and this should be carried out at a regular interval. Okay, and we can say that if there is any changes in the premises, either in the process, either in the material, okay, either any methods, either any equipment, any utilities, then there should be the validation process should be carried out. For at every during major changes, there is the need of the validation. Okay, now let's just begin with the types of the validation. How many types of validation are there? Mainly there are very types, but mainly we will be discussing about the 4 types. Okay, firstly, process validation, cleaning validation, and then we will go about the equipment validation. Equipment validation mainly named as the qualification, and then we will be discussing about the revalidation. When should we revalidate the processes? Okay, then let's begin with the process validation. Okay, what is process validation? We will be discussing about the process validation in a very detailed way. Okay, as per the US FDA has proposed that process validation is defined as the evidence, as the documentary evidence, authorized documentary evidence that will assure a high degree in a high degree way that the products are produced in a very qualitative way. Okay, that the process will produce the products consistently in a very qualitative way, in a very specified way that will assure its quality. Okay, as per the US FDA, that process validation can be carried out in 3 stages. Firstly, one, process design, and then process qualification, and then after continued process verification. Okay, Firstly, process design. Process design, in this process design we will gain the information about the commercial information as per the development and the scale of process. Okay, and then in stage process qualification. Process qualification stage will confine that the process design is capable of producing the reproducible commercial batch. And then after that, stage 3, continued process verification. Okay, continued process verification will be assuring that the ongoing reproducible production of the commercial batches are assured of quality. We have assured that quality, okay, that is in a state of control. Quality is in a state of control. So these steps are carried out as a process validation. Okay, under process validation there are mainly 4 types. Okay, firstly we will be discussing about some important things, and then we will be much more clear about the types. For types, we will be discussing about the prospective validation, okay, concurrent validation, retrospective validation, and then we will discuss about the revalidation. Okay, for example, firstly we should take an example like when a pharmaceutical manufacturing company established, it have very departments, it have many departments. Okay, storage, QC, QA, production, and other administrative department as well. Okay, when a company says that they need to produce or they need to manufacture a drug, then what happens? Then they will give that information to the R&D department, and then R&D department will say that okay, and they will accept the proposal, and then R&D department will begin its activities. They will choose the API and excipients and process as per the written protocols and the guidelines. Okay, they will set up every conditions, every methods. Okay, and they will take the 3 formulations. First 3 formulations are taken by the R&D, and then after that 3 formulation production, they will give that for 3 formulations to the QC department. And when QC department analyzes that 3 formulations, if that 3 formulations pass, okay, as per their protocol and the guideline or the process they have mentioned, then that report is sent to the R&D. And when that 3 batches are when that 3 formulations are passed, then that R&D will be applying to the DDA. In context of Nepal, they will submit the 0-month analysis report of the raw material, of the finished product, raw material specification, finished product specification. Okay, and R&D tentative BMR. R&D will give the tentative BMR as well. They will provide the hard copy, as well as they will fill up this the software through the software like DAMS. They will fill up the process for the application, for the apply of the manufacturing license of that drug to the DDA. Okay, and after that they will wait for the result. I mean, they will wait for some intervals like 3 months and 6 months. They need to provide the 3 month and 6 month real and accelerated stability testing report of that 3 batches. Okay, or of that batches. And then we all know that in real accelerated we need to analyze the data at different temperatures and the humidity. In case of real, 30 degree plus minus 2 degrees Celsius temperature and humidity 75 plus minus 7, while in accelerated 40 degree plus minus 2 degrees Celsius temperature and humidity 75 plus minus 5 degrees. Okay, and in both cases, if that R&D gets the past report of that 3 month and 6 month accelerated testing, stability testing, then R&D will submit that report, stability report, to the DDA, and then DDA will provide the manufacturing license to the pharmaceutical manufacturing company about that drug. Okay, after receiving that marketing license, only the company will begin the production in commercial batches, production of the commercial batches in its production department, otherwise they can't begin. Okay, and in this case, in case if the test fails, then what happens? Okay, if the stability testing reports fails, then the R&D will take the another formulations and they will give these supportive documents to the DDA, and they will say about the revert the document. Okay, and after this process of analysis, or the way the steps are validated, or the documented evidences are provided, okay, due in the R&D stage, is called as the prospective validation, where it's called as the prospective validation. And when after receiving the marketing license from the DDA, the production starts in the commercial batches in the production stage, then that stage, okay, the protocols which have been designed by the R&D, in that way the commercial production begins. Then the steps of control, okay, and during the production stage, and the testings are done in the QC as per the protocols or the guidelines set by the R&D, then that is called as the concurrent validation. Okay, this is called as the concurrent validation. And after this, when the commercial batches pass every test as per the protocols and the guidelines of pharmacopoeias, okay, then the market, okay, then the product will release in the market. And after a regular interval of time in real and accelerated conditions, the historical analysis of the batches, okay, the historical analysis of the commercial batches will be carried out, and this is called as the retrospective validation. Okay, means in the retrospective validation, we will take the batches, okay, commercial batches, we will take the samples of the commercial batches, and the samples were already kept in the stability chamber in real and accelerated conditions, and we will take the sample from that, and then we will study the historical data of that commercial batches of different batches. Okay, and then we'll give the report, then that is called as the retrospective validation. And when we should do the revalidation process, revalidation is carried out mainly when there is major changes either in the formulations, either in the premises, equipment, facilities, or any other serious degradation occurs in the drugs, or any serious side effects are seen in the market when the products were released in the market, then revalidation is required. Okay, and in this case we will be going through every steps. Revalidation is required when there is new changes. Okay, and if there is no new changes in stability or in other things as well, but also we need to carry out the validation process at every 5 years. This is the concept of the validation process. Okay, and then this is the mainly process validation. Okay, and what happens in the another type of validation, like this cleaning validation. Okay, now we will start about the cleaning validation. What is cleaning validation? Okay, cleaning validation is a procedure of establishing the evidence that cleaning processes for manufacturing equipment prevents the product contamination. Okay, for example, when product A is produced in the equipment, then after production of the product A, we need to clean the equipment, and then only the equipment will be ready for the production of the another product, like product B. Okay, firstly, after the manufacture of the product A, we need to clean that equipment, and we will test whether that equipment is cleaned or not. Okay, we can use various methods like as rinse method, swab method. Okay, and after the test is passed, after we find that the equipment is passed, okay, the cleaning validation equipment has passed the cleaning validation, then only the manufacture of the product B will begins. Okay, not more than 10 ppm of the product A should be present on the equipment before the production of the product B. Okay, mainly the main purpose of the cleaning validation is to prevent the product contamination. Cleaning validation has to prevent product contamination, so it's a very much important method of validation. While another validation like as the equipment validation, which is mainly termed as the qualification. Okay, as I have already discussed in the qualification. Okay, qualification is a part of the validation, and in qualification we had discussed about the DQ, IQ, OQ and PQ. Okay, qualification is a part of the validation, but qualification is not only limited to the process. It has four types. Okay, it can be mainly described in the four types. You can view my that you can see that video. Okay, and if you have any confusion, then please let me know. Okay, now we will discuss about the AMV, analytical method validation. Okay, the process by which it is established by the laboratory studies that the performance characteristics of the method meet the requirements for the intended analytical application. This is called as the analytical method validation. Okay, under analytical method validation we will study the following things like accuracy, precision, okay, specificity, and then limit of quantitation, range, robustness, okay, and linearity and limit of detection. These things are studied in detail. Okay, we'll study in detail in further video, and for now I think our video is over. And if you people have any queries, then please let me know, and if you people want a video on any other topics, then please let me know. And so much, thank you so much. Bye bye.